separate things, they're actually using the same basic genetic language.
The same playbook.
Exactly. The same molecular pathways, the same machinery at the core. So our plan today is to walk you through this material really methodically. We want to make sure we cover it all clearly, step by step, showing you those deep connections.
Okay, and we've got a few handy metaphors to guide us right now. First up,
Mm.
development. It's described as this precise choreography of sulfate.
Mm-hmm. An incredibly complex dance. Billions, trillions of cells, all knowing exactly where they need to go, what job they need to do, and when. It's astonishing, really.
Then we have stem cells. These are the patient architects of renewal.
Yeah, they're like the reserve workforce.
Yeah.
Holding on to that potential to rebuild, to repair tissues. They sit quietly in these specific spots, these niches.
Waiting to be called up.
Until they get the signal, yes.
And finally, the dark side. Cancer. This is where that beautiful developmental dance,
Yeah.
well, it forgets when to stop dancing.
Right. The music keeps playing, gets way too loud, becomes chaotic, destructive. So we're going to start right at the beginning, tracing that initial genetic cascade, how the very first blueprint gets laid down. Okay, to really get how complex we are as adults, you have to go back to the start, that genetic cascade that builds the foundation. And for you listening, we're going to track this using a little mnemonic. Mothers give pair some, MGPS.
Right, helps keep it orderly. Let's start with M, the maternal effect genes. It's kind of mind-blowing that the very first instructions don't even come from the embryo's own DNA.
It really is. These genes, they're expressed by the mother while she's forming her eggs. That process is called oogenesis. And the actual products, the mRNA or the proteins, they get physically deposited right into the egg cytoplasm before fertilization.
Wow. So the mother literally packs the instructions into the egg beforehand, setting the stage.
Exactly. Their job is absolutely critical for that very early development, especially setting up the main body axes, front to back, top to bottom. And this happens before the embryo's own genes, the zygotic genome, really kick in.
Okay, so they establish the fundamental coordinates. Are there classic examples?
Oh, absolutely. The best ones show this gradient idea. You have the mRNA for a gene called bicoid. It gets concentrated at one end, the anterior pole.
The head end.
Right, and it controls head formation. Then at the other end, the posterior pole, you find nanos mRNA.
Which controls the abdomen, the tail end.
Precisely. These opposing gradients, high bicoid at the front, high nanos at the back, they set up that fundamental anterior-posterior polarity. Everything else builds on that initial asymmetry.
Okay, so maternal genes set the main map. Then the embryo's own DNA starts working. That brings us to G in our mnemonic gap genes.
Right. Now we're talking about zygotic genes, the embryo's own, and they're switched on by those maternal gene products we just discussed.
And what's their role? They're called gap genes.
They define the first really large territories of the embryo. Think of them as marking out broad, overlapping bands along the body axis, sort of dividing the whole thing into maybe 3 or 4 major zones.
And the name gap.
It comes from what happens if they're mutated. If you lose a gap gene, you don't just lose a tiny bit. You lose whole chunks, entire groups of adjacent body segments. It leaves a literal gap in the body plan.
Okay, so big regions established. Then we need more detail. That's PRO, the pair rule genes. If gap genes made the big zones, what do pair rule genes do? Slice them up.
That's a perfect way to put it. They slice the territories. They respond to those broad, overlapping gap gene bands. And they introduce periodicity. They paint stripes.
Stripes, okay.
Yes, they divide those big regions into precisely spaced stripes. These stripes are the direct precursors of the body segments. And this step creates, specifically, 14 parasegments. Genes like even skipped and Fushi-Tarazu are key players here, making sure that striped pattern emerges correctly.
14 stripes laid down. We're getting more refined. What's the last step in segmentation? Segment polarity genes.
Right. These come a bit later, after the embryo has actually formed individual cells after cellularization. And that timing is important.
Why?
Because now cells can talk directly to their neighbors. These genes rely on cell-to-cell signaling. Their job is to sharpen the edges. They define the front half, anterior, and the back half, posterior, of each of those 14 segments, giving each stripe an internal directionality.
Okay, so they add detail within each stripe. Examples.
Key ones are engrailed and wingless. And notice that name, wingless. That immediately points to the once signaling pathway.
Which we know is super important later on in stem cells and, unfortunately, in cancer.
Exactly. The same players keep showing up. It really underscores that shared vocabulary we talked about.
All right, so the segmentation cascade mothers give pairs. Some has physically laid out the segments. They exist. But what do? What tells one segment to become part of the head and another to become part of the thorax with legs?
That's precisely the job of the next set of master regulators,
Hmm.
the homeotic genes, or as they're usually called, the Hox genes.
Okay, Hox genes. They give each segment its unique ID.
That's it. If the segmentation genes built the compartments on the train, the Hox genes label each compartment. This is the engine car, this is a passenger car, this is the dining car. They assign the specific identity.
And if they mess up,
You get what's called homeosis, body parts developing in the wrong place, like legs growing where antenna should be on a fly. It shows their power in defining regional identity.
what makes a Hox gene a Hox gene? Is there a specific structure?
Yes, the defining feature is a highly conserved DNA sequence about 180 base pairs long called the homeobox.
The homeobox. Okay.
This homeobox sequence codes for a special DNA binding part of the protein called the homeodomain. So Hox genes essentially produce master switch proteins, homeodomain transcription factors, or TFs that control hundreds of other downstream genes, the ones that actually build the leg or the wing.
And there's something truly amazing about how these Hox genes are organized, isn't there? The collinearity principle. Can you break that down? It sounds fundamental.
It is absolutely fundamental and, yes, mind-blowing. Collinearity means that the physical order of the Hox genes along the chromosome directly mirrors the order in which they are turned on, both in space along the body axis and in time during development.
Wait, so the genes at one end of the chromosome cluster control the head region, and the genes at the other end control the tail region.
Exactly. Genes at the 3' bend of the cluster are expressed earlier and more anteriorly towards the head. Genes progressively further towards the 5' end are expressed later and more posteriorly towards the tail.
It's like the body plan is literally mapped out sequentially on the chromosome.
It is. This linear arrangement ensures the body pattern unfolds in a precise, ordered sequence from front to back. It's an incredible link between genome structure and organismal structure.
And this isn't just flies, right? Humans have these too.
Oh, absolutely. Humans have 4 clusters of Hox genes, Hox A, B, C, and D. They are absolutely essential for patterning complex structures like our vertebral column, our spine, and our limbs.
So it follows that mutations would cause serious problems.
Directly. The sources are clear. Mutations in human Hox genes lead to severe limb abnormalities and vertebral defects. Messing with these master identity genes has profound consequences for our skeletal structure. The blueprint analogy is very literal here. Okay, so we have the genes setting the plan, assigning identities. But how does that genetic information translate into actual cell behaviors? How do cells know what to do based on their location? This relies heavily on communication.
Through signaling molecules.
Precisely, particularly powerful ones called morphogens. These are secreted molecules that spread out, forming a concentration gradient across the tissue.
Right, and this leads to the threshold model, which sounds simple but is really powerful.
It is. The core idea is that a cell's fate, what it decides to become, depends on the concentration of the morphogen it experiences. Different concentration levels trigger different responses.
Like a high dose tells it to be one thing, a medium dose something else, and a low dose maybe a third thing.
Exactly. It's dose-dependent specification. If the concentration crosses a certain threshold, it flips a switch inside the cell, leading to a specific fate. If it crosses a higher threshold, it flips a different switch. It allows for complex patterns to emerge from relatively simple gradients.
And we absolutely need to go through the major signaling pathways involved here because, as you keep saying, these are the very same systems that get hijacked in cancer.
We do. Understanding their normal developmental role is key to understanding how they cause disease when deregulated. Let's start with a really famous one, Sonic Hedgehog.
Shh. Okay, shh. Sounds friendly. What does it do?
It's vital. It plays a huge role in patterning the ventral side, the underside of the developing neural tube, and it's also essential for organizing the digits on our hands and feet.
And the mechanism. How does it work?
It involves a receptor called patched, or PTC. Normally, PTC keeps another protein called smoothened, or SMO, inhibited. It keeps it quiet. But when shush comes along and binds to patched, it relieves that inhibition. Smoothen becomes active. And activated, Smoothen then triggers a cascade involving the GLI family of transcription factors, which go into the nucleus and turn on target genes.
So binding basically releases the brake on smoothened.
That's the core of it. Patched is the brake, releases it, Smoothened hits the gas, GLI drives the car.
Got it. And developmental problems if this pathway is faulty.
They can be devastating. Errors in signaling are linked to holoprosencephaly,
Which is?
a severe birth defect where the forebrain fails to divide properly into 2 hemispheres. It underscores sh's role in fundamental brain patterning. On the flip side, too much signaling can cause polydactyly, the formation of extra fingers or toes. Shows its role in limb patterning, too.
Right, balancing the signal is critical. Okay, next up, windlass. We already met this in segment polarity. What's its broader role?
Wnt is huge. Developmentally, it's crucial for establishing the anterior-posterior axis, and it's absolutely essential later in life for maintaining stem cell populations, particularly in tissues that turn over rapidly, like the gut.
And its mechanism involves a protein called beta-catenin, right?
Yes, Bonhatten is the star player. Normally, in the absence of a Wnt signal, Bonhatten is constantly being targeted for destruction by a protein complex, often called the destruction complex, which includes a key protein called APC.
Okay, so bond-catenin is usually kept at low levels.
Right. But when Wnt binds to its receptor complex at the cell surface, it triggers a signaling cascade that inactivates that destruction complex.
Ah, so bond-catenin is no longer destroyed.
Exactly. It accumulates, it stabilizes, and then it travels into the nucleus where it partners with TCFL-EF transcription factors to switch on Wnt target genes, often related to proliferation and stemness.
And this pathway has a really strong direct link to a major cancer, doesn't it?
Tragically, yes, colon cancer. The vast majority of hereditary colon cancers, and many sporadic ones, involve mutations that cripple that destruction complex, very often in the APC gene itself.
So if APC is broken,
Then B-catenin is always stable, always active, even without any Wnt signal. It's constantly telling the cell to proliferate, driving tumor formation. It's a classic example of a developmental pathway hijacked.
wow. Okay, moving on. BMP-TGF pathway.
This family of signaling molecules, including bone morphogenetic proteins, BMPs, and transforming growth factor beta, TGFA, signal primarily through the phosphorylation of intracellular proteins called SMADs.
SMADs.
Okay.
And their role.
They are critical for setting up the dorsal ventral axis, the top to bottom axis in the early embryo, and they continue to be important throughout life in bone formation, cartilage development, and connective tissue homeostasis. Defects here often lead to various skeletal dysplasias, problems with bone growth and structure.
Makes sense. Next,
Yeah.
FGF, fibroblast growth factor.
Yeah, FGFs are potent drivers of cell proliferation and differentiation, crucial for big events like limb outgrowth and brain development. They signal through a class of receptors we'll hear a lot about, receptor tyrosine kinases, or RTKs.
RTKs.
Yeah.
These are major players, right?
Absolutely. When FGF binds, the RTK receptors pair up, activate their intrinsic kinase activity, and trigger complex downstream signaling cascades, common ones being the MEPK pathway, driving proliferation, and the PI3K pathway, promoting survival.
And developmental disorders linked to FGF.
Mutations affecting FGF receptors are frequently linked to craniosynostosis,
Such as?
the premature fusion of the bony plates of the skull. This can restrict brain growth and cause abnormal head shapes. It highlights FGF's role in coordinating skeletal development.
Okay, then there's retinoic acid. This one seems a bit different.
It is. Retinoic acid isn't a protein. It's a small molecule, a metabolite of vitamin A. And it doesn't use the surface receptor. It diffuses into the cell and binds to nuclear receptors that act directly as transcription factors.
And its role.
It's very important for anterior-posterior patterning, particularly in the developing central nervous system and limbs. But the really critical thing the sources emphasize about retinoic acid is,
Its danger.
yes, it's a potent teratogen. That means exposure to incorrect levels during pregnancy can cause severe birth defects. Things like Accutane, the acne medication, which is a retinoic acid derivative, carry very strong warnings for pregnant women. It shows how exquisitely sensitive development is to the levels of these signaling molecules. Too much or too little can be catastrophic.
A very narrow window for proper function. Okay, one more key pathway for early development. Nodal.
Nodal is the primary driver for establishing left-right asymmetry in the body.
Left-right asymmetry. You mean making sure the heart is on the left, the liver on the right?
Exactly that. It's a fundamental patterning event. Nodal signaling, which also works through SMAD proteins, specifically SMA23, sets up a cascade that determines the sidedness of our internal organs.
And if it fails?
Failure in the nodal pathway can lead to conditions like situs inversus, where the arrangement of the internal organs is a mirror image of normal, or even more complex randomization of organ placement called heterotaxy. It's a fascinating example of how development creates not just pattern, but specific 3D organization.
Okay, we've covered the initial genetic blueprint and the major communication lines, the signaling pathways that orchestrate development.
Yeah.
But building a complex organism isn't just about turning genes on at the right time. It's also about keeping them off permanently in cells where they don't belong.
Exactly. You need stability. You need memory. A liver cell needs to stay a liver cell for decades. That long-term stability, that developmental memory, is largely encoded not in the DNA sequence itself, but in how the DNA is packaged. And that's the realm of epigenetics.
Right. But before we dive deep into epigenetics, let's quickly revisit three of those signaling pathways we just discussed, but specifically look at how they get corrupted in cancer, because they are prime examples of developmental systems going rogue. Hashtag TechTechA. The control systems and communication,
Okay, let's start with Notch delta. We mentioned its role in development. How does it work again?
it needs direct cell contact, right? The delta protein on one cell binds to the Notch receptor on the cell next door.
Correct. And that binding event triggers enzymes to actually cleave the Notch receptor. Part of it breaks off.
The intracellular part.
Yes, the Notch intracellular domain, or NICD. This piece then travels to the nucleus and teams up with transcription factors like CSL to turn on Notch target genes.
And its developmental job is often about choices, like lateral inhibition.
Precisely. Like in the developing nervous system, one cell decides to become a neuron and uses Notch signaling to tell its immediate neighbors, don't become neurons, choose a different fate. It helps create patterns and boundaries. It's also involved in blood vessel formation, angiogenesis.
And the cancer link.
When Notch signaling gets stuck in the on position, often due to mutations in the receptor itself, it drives certain cancers. It's particularly strongly implicated in T-cell acute lymphoblastic leukemia, TALL. Uncontrolled Notch activity keeps those T cells proliferating and prevents them from differentiating properly.
Okay. What about the hedgehog pathway's dark side? We covered the Schiff mechanism.
Right. Spine's patched, releases smoothened, activates GLI. In cancer, the system often gets jammed on without needing the Schiff signal. You might get a mutation in patched so it can no longer inhibit smoothened, or a mutation in smoothened itself that makes it permanently active.
So constant GLI activity.
Constant GLI activity, driving expression of genes that promote cell survival, proliferation, and self-renewal. This constitutive activation is a hallmark of basal cell carcinoma, the most common type of skin cancer, and also some brain tumors like medulloblastoma.
Makes sense. And then the big ones.
Yeah.
The receptor tyrosine kinases, RTKs. This includes receptors for signals like FGF, EGF, epidermal growth factor, VEGF, vascular endothelial growth factor.
Yes, these are major engines for cell growth, survival, and proliferation. As we said, ligand binding causes them to dimerize pair up, which activates their kinase domain. They then phosphorylate themselves and other proteins, kicking off those huge intracellular signaling cascades,
Like Razumab PK
driving proliferation,
and PI3 KPET.
promoting cell survival and growth. These pathways are absolutely central to normal development, but they are probably the most frequently mutated or amplified pathways in human cancer.
We hear about specific examples all the time in cancer treatment.
Absolutely. Think of HER2, which is an EGF receptor family member, being overexpressed or amplified in many breast cancers, or mutations in the EGFR, epidermal growth factor receptor, itself driving a significant fraction of lung cancers. Targeting these specific RTKs, or the pathways they activate, is a cornerstone of modern cancer therapy.
Okay. Now, development isn't just chemical signals. Cells need to stick together, and sometimes they need to move. Let's talk about adhesion. What holds tissues together?
The primary glue, especially in epithelial tissues like skin or the lining of the gut, are the cadherin molecules. These are transmembrane proteins that link cells to their neighbors in a calcium-dependent way. They form strong cell-cell junctions.
And the sources highlight one cadherin in particular.
E-cadherin. It's considered the guardian of the epithelial state. Its presence is crucial for maintaining the integrity and structure of epithelial sheets, and its loss, well, that's a pivotal event in cancer progression.
Why is losing E-cadherin so critical?
Because losing E-cadherin is often the key initiating step for a process called epithelial to mesenchymal transition, or EMT.
EMT, we'll come back to that, but basically the cell changes its character.
Dramatically.
Yeah.
It loses its tight connections, its fixed position, its epithelial characteristics, and it gains migratory abilities. It becomes mesenchymal-like. And that transition is absolutely required for cancer cells to break away from the primary tumor and metastasize to spread.
So loss of E-cadherin unlocks the door for metastasis. What about connecting cells to their surroundings, not just to each other?
That's the job of the integrins. These are another class of transmembrane receptors, but instead of linking cell to cell, they link the cell's internal cytoskeleton to the extracellular matrix, ECM, the scaffold of proteins and sugars outside the cell.
So they're like the cell's feet, grabbing onto the environment.
That's a great analogy. They anchor the cell, but they also act as sensors. They transmit signals both ways from the outside ECM in to tell the cell about its environment. Is it stiff? Is it soft? What proteins are present? And from the inside out to control adhesion and migration. They're crucial for cell movement and survival signaling.
Can we see a developmental process where this kind of adhesion change and migration is normal and essential?
The classic, beautiful example is the neural crest.
Ah, yes. Tell us about them.
Neural crest cells are a remarkable population. They originate early in development along the top edges of the forming neural tube. But they don't stay there. They undergo a temporary EMT, lose their E-cadherin, become migratory, and then embark on these long journeys throughout the developing embryo.
And they become many different things.
A huge variety. They differentiate into neurons and glia of the peripheral nervous system, pigment cells, melanocytes in the skin, smooth muscle cells, bone and cartilage cells, especially in the face and skull. It's a stunning example of controlled, developmentally programmed migration and differentiation using mechanisms like EMT for constructive purposes. Okay, so now we get to the core of that developmental memory, epigenetics. How does a cell lock in its fate? The first major mechanism is DNA methylation.
Right. This involves adding a chemical tag, a methyl group, directly onto the DNA molecule itself, specifically onto cytosine bases, usually where they're next to a guanine, the CpG dinucleotide.
Correct. And there are specific enzymes that do this. The DNMTs, DNA methyltransferases. DNMT3A and DNMT3B are the de novo methyltransferases. They establish new methylation patterns during development, writing the epigenetic code.
And DNMT1.
DNMT1 is the maintenance methyltransferase. When the cell divides and replicates its DNA, DNMT1 recognizes the methylation pattern on the old strand and copies it onto the newly synthesized strand. This ensures the epigenetic marks are faithfully passed down through cell divisions.
So it preserves the memory. And how is methylation removed if needed?
That's primarily done by the TET enzymes, which initiate a process leading to demethylation, erasing the marks.
But functionally, what does adding these methyl groups do to a gene?
Primarily, it silences it. When the promoter region of a gene, especially CpG islands within it, becomes heavily methylated, it tends to recruit proteins that condense the chromatin structure, making it physically difficult for the transcription machinery to access the gene. It's a very stable OFF switch.
And the body uses this for important jobs.
Absolutely. It's the mechanism behind X chromosome inactivation in females, ensuring dosage compensation. It's used for genomic imprinting, silencing one parental copy of certain genes, and crucially for our discussion, it stabilizes cell lineages by permanently shutting down genes that are specific to other developmental pathways. It helps lock in cell identity.
You mentioned a reset of these marks.
Yes, there's a fascinating, massive wave of demethylation right after fertilization, essentially wiping the epigenetic slate clean for the zygote. Then, as the embryo develops, particularly during gastrulation when the fundamental germ layers form, there's a huge wave of de novo methylation, rewriting the epigenetic landscape to establish the initial lineage commitments.
And the cancer connection.
Just profound. While cancer involves genetic mutations, it also heavily involves epigenetic alterations. A common finding in tumors is aberrant hypermethylation, excessive methylation, specifically at the promoters of tumor suppressor genes.
So the cancer cell uses methylation to silence the very genes that should be stopping its growth.
Exactly. It hijacks this normal silencing mechanism to turn off its own brakes. This is just as important as genetic mutation in driving cancer.
Okay, methylation is one layer. What about modifications to the proteins that package the DNA, the histones? That's chromatin modification.
Right. If DNA methylation is writing on the DNA itself, histone modification is like writing on the spools the DNA is wound around. And there are many different types of modifications, creating a complex histone code.
What are the main ones we should know?
Two key ones are acetylation and methylation, but this time, histone methylation, not DNA methylation. Let's start with acetylation.
Okay, adding an acetyl group.
Yes, by enzymes called histone acetyltransferases, HATs, and removal by histone deacetylases, HDACs. Acetylation generally neutralizes the positive charge on lysine residues in the histone tails.
And what effect does that have?
It loosens the interaction between the histones and the negatively charged DNA. This causes the chromatin structure to relax, to open up. Open chromatin generally means genes are accessible and can be transcribed, so acetylation is typically associated with active genes.
Makes sense. Open, on. What about histone methylation?
Histone methylation is more complex. Enzymes called histone methyltransferases, HMTs, add methyl groups, and lysine demethylases, KDMs, remove them. But unlike acetylation, methylation doesn't always mean the same thing.
How so?
It depends on which amino acid residue on the histone gets methylated and how many methyl groups are added, mono, diol, or trimethylation. For example, trimethylation of lysine 4 on histone H3, written as H3K4Me3, is a strong mark of an active gene promoter.
Okay, H3K4Me3 kind of active.
But trimethylation of lysine 27 on histone H3K27E3 is a major mark of gene repression. It's associated with silenced genes and condensed chromatin.
So methylation can mean on or off, depending on the context. H3K27Me3 equals repressed.
Exactly. It's a more nuanced code than acetylation.
This complexity leads to a really interesting state found in stem cells, right? Bivalent domains.
Yes. This is fascinating. In embryonic stem cells, ESCs, many key developmental genes have both the activating H3K4Me3 mark and the repressive H3K27E3 mark present at their promoters simultaneously.
Both on and all off-F signals at the same gene. How does that work?
It means the gene isn't fully active, nor is it fully stably silenced. It's held in a poised state, ready to go either way quickly.
Why would that be useful?
It allows ESCs to respond rapidly to differentiation cues. If the signal says activate this gene, the repressive H3K27E3 mark can be quickly removed, leaving the activating H3K4Me3, and the gene switches on in. If the signal says silence this gene, the H3K4Me3 mark is removed, locking in the repression via H3K27E3. It provides developmental flexibility.
Very clever. Now, all these marks on DNA and histones influence how tightly packed the chromatin is. But you also need machinery to physically move things around, right?
Yeah.
To expose or hide genes.
Absolutely. That's the job of chromatin remodelers. These are large protein complexes, like the well-known SWA-SM complex.
Swiss and F. What does it do?
Think of them as molecular bulldozers. They use the energy from ATP hydrolysis to physically slide nucleosomes along the DNA, or even lift them all together. This action can expose promoter sequences to allow transcription factors to bind, or conversely, it can condense regions to silence them. They control the physical accessibility of the genome.
And their importance is highlighted
By a really striking statistic from the sources, components of the Swiss and F complex are mutated in over 20% of all human cancers.
20%. That's huge.
It is. It tells you that controlling the physical landscape of the genome is absolutely critical for preventing malignancy. Losing that control is a major driver of cancer.
Okay, so we have these epigenetic marks, methylation, histone modifications, creating states like active, repressed, or poised. But how are these states maintained over the long term, especially the repressed state that locks in cell identity?
That involves a constant battle, a dynamic tug-of-war between two major groups of regulatory proteins, the polycomb group and the trithorax group.
Polycomb versus trithorax. Okay, who does what?
The polycomb group proteins, PCG, are the master silencers. Their main job is to maintain the repression of developmental genes in differentiated cells, ensuring they stay off when they shouldn't be active. For instance, keeping Hox genes that specify leg turned off in a brain cell.
How do they silence?
A key polycomb complex is PRC2, polycomb repressive complex 2. Its enzymatic subunit is EZH2. And what EZH2 does is catalyze the addition of that repressive histone mark we just talked about, H3K27Me3. So PCG proteins essentially paint genes with the OFF signal.
Got it. Polycomb silencers, H3K27Me3 equals OFF. Who's on the other side of the tug-of-war?
The trithorax group proteins, TRXG, they are the activators. Their job is to maintain the active state of genes that should be expressed in a particular cell type. They counteract the silencing activity of polycomb.
And how do they activate?
One key mechanism is adding the activating histone mark, H3K4Me3. So trithorax proteins maintain the on-end signal. The long-term fate of a gene, whether it stays on or off in a lineage, depends on the balance of power between polycomb and trithorax activities at its locus.
A constant battle maintaining cell identity. Fascinating. Beyond these big protein complexes, we're also learning more about regulatory roles for RNAs that don't code for proteins, right? Non-coding RNAs.
Yes, they are emerging as major players in gene regulation, including epigenetic control. We already mentioned one classic example, the long non-coding RNA, LNCRNA, called XC.
Right, for exon activation.
Exactly. XC RNA physically coats the entire inactive X chromosome in female mammals and recruits polycomb complexes and other silencing machinery to shut down almost all the genes on that chromosome. It's a large-scale epigenetic event mediated by a non-coding RNA.
Are there other examples?
Yes, the sources mention another LNCRNA called HOTAIR. HOTAIR acts as a scaffold, binding to specific DNA regions and also recruiting polycomb machinery, thereby targeting specific genes for silencing in different tissues. It can act across chromosomes, which is quite remarkable.
Yeah, and smaller RNAs, microRNAs.
MicroRNAs, miRNAs, typically regulate gene expression post-transcriptionally, often by degrading target mRNAs. The example given is miR145, which is known to promote differentiation. Importantly, miR145 levels are often found to be low or suppressed in stem cells.
Why would that be?
Because keeping miR145 low helps prevent differentiation, thus maintaining the pluripotent or stem-like state. It shows how non-coding RNAs are integrated into these networks controlling cell fate.
Okay, so we have DNA methylation, histone mods, polycontrathorax, non-coding RNAs, all contributing to epigenetic memory. What happens when this memory system makes mistakes, particularly with those genes that are supposed to remember which parent they came from? This leads to imprinting disorders.
Exactly. Genomic imprinting is that phenomenon where only one copy of a gene, either the one inherited from the mother or the one from the father, is expressed, while the other copy is epigenetically silenced, usually by DNA methylation. Imprinting disorders occur when this specific parental silencing goes wrong.
Can you give us some examples?
The classic pair involves a specific region on chromosome 15, specifically 15q1113. If the paternal copy of this region is lost or deleted or incorrectly silenced, it results in Prader-Willi syndrome,
Prader-Willi.
associated with.
Characterized by developmental delay, hypotonia, low muscle tone in infancy, and later hyperphagia leading to severe obesity.
Okay. And what if the problem is with the maternal copy in that same region?
If the maternal copy is lost or mutated, specifically involving the UBE3A gene within that region, it leads to a completely different disorder, Angelman syndrome.
Angelman syndrome features.
Also involves developmental delay, but typically includes seizures, ataxia, movement problems, and a characteristic happy demeanor with frequent laughter. It's striking because the exact same chromosomal region is involved, but the outcome depends entirely on which parent's contribution was lost.
A stark illustration of imprinting. Are there others? Yes, involving growth regulation. Beckwith-Wiedemann syndrome is an overgrowth syndrome. Larger body size, enlarged organs, increased risk of childhood cancers. It's often caused by problems with imprinted genes on chromosome 11, particularly leading to biallelic expression from both parental copies, of the potent growth factor IGF-2, which is normally only expressed from the paternal copy.
Too much IGF-2 leads to overgrowth. What about the opposite?
The opposite is Silver-Russell syndrome, which causes growth restriction, small size at birth, slow growth. This is often linked to the opposite problem at the same chromosome 11 region, specifically hypomethylation, reduced methylation, meaning potential activation, of the normally silenced maternal copy of H19, or loss of methylation at the IGF-2 locus on the paternal chromosome. Again, it's all about currentine H19 IGF-2 imprinting control region on chromosome 11p15, leading to reduced IGF-2 levels or other growth disturbances.
So these syndromes really hammer home how critical it is not just to have the right genes, but to have them correctly marked with their parental origin through epigenetics.
Absolutely. Epigenetic memory is essential for normal development.
Okay, we've journeyed through the initial construction, the choreography of development. Now let's shift focus to how the structure is maintained throughout life. This brings us to those patient architects of renewal, our stem cells.
Right. Stem cells are the reservoir for replacing cells lost to normal turnover, injury, or disease. Their defining characteristics are self-renewal, the ability to divide and make more stem cells, and differentiation, the ability to produce specialized cell types.
And we classify them based on how much differentiation potential they have, right? Their potency. Hashtag A.
Yeah.
Defining potency, the
Yes.
spectrum.
Exactly. Potency exists on a spectrum. At the very top, the most potent cells are totipotent.
Toto meaning all. What can they form?
Literally everything. A totipotent cell can differentiate into all possible cell types, including the specialized cells of the placenta and other extra embryonic tissues needed to support development. The zygote, the fertilized egg, and the cells from the first couple of divisions are totipotents. They can, in principle, form an entire organism on their own.
Okay, maximum potential. What's the next level down?
Pluripotent. Pluri meaning many.
Like embryonic stem cells.
Exactly. Embryonic stem cells, ESCs, derived from the inner cell mass of the blastocyst stage embryo are the classic example. Pluripotent cells can differentiate into all cell types of the three primary germ layers, ectoderm, mesoderm, and endoderm, which means they can form virtually any cell type in the body proper.
But not the placenta.
Correct. They cannot form the extra embryonic tissues like the placenta. So they can't form a complete organism alone, but they have immense potential for generating tissues. These cells are characterized by the expression of key master transcription factors like OCT4, SOX2, and NANOG, which maintain their pluripotent state. They are the focus of much regenerative medicine research.
Right. Now, most stem cells in our adult bodies aren't pluripotent, are they? They're more restricted.
That's right. Most adult stem cells are multipotent. Multi meaning several.
So tissue-specific.
Yes, they are generally committed to producing the cell types of a specific tissue or organ system. The classic example is the hematopoietic stem cell, HSC, found in bone marrow. HSCs, which often express the surface marker CD34, are responsible for generating all the different types of blood and immune cells, red blood cells, white blood cells, lymphocytes, neutrophils, etc., platelets, but they can't make liver cells or neurons.
Okay, multipotent.
Yeah.
Restricted to one tissue family. Can we get even more restricted?
Yes. Below multipotent, you have oligopotent cells, oligo meaning few. These stem cells can differentiate into just a few related cell types. For example, within the hematopoietic system, you have lymphoid progenitor cells, making lymphocytes, and myeloid progenitor cells, making red cells, granulocytes, etc. They are more restricted than the HSC they came from.
And the final step.
Unipotent. Uni meaning one. These cells can typically only produce one specific type of differentiated cell, along with renewing themselves. An example might be the stem cells in the epidermis that generate keratinocytes, the main cell type of the skin. They ensure the constant renewal of that single lineage. #TAG-B embryonic and induced pluripotency.
Let's talk more about those embryonic stem cells, ESCs. They come from the inner cell mass of the blastocyst. How are they kept in that undifferentiated pluripotent state in the lab?
It requires very specific culture conditions designed to mimic the signals within the early embryo that prevent differentiation. Central to this is maintaining the high expression levels of those core pluripotency factors, OCT4, SOX2, and NANOG.
And growth factors are needed too.
Yes. The specific factors depend on the species. For mouse ESCs, a key factor is leukemia inhibitory factor, LIF, which signals through the STAT3 pathway. For human ESCs, maintaining pluripotency often relies more on pathways activated by factors like FGF2, fibroblast growth factor 2, and activin nodal. Essentially, you're actively suppressing differentiation pathways.
Of course, ESCs have always come with significant ethical debate due to their origin.
Indeed. The derivation of ESCs typically involves the destruction of a human embryo, which raises major ethical concerns for many people. This was a huge barrier to their potential therapeutic use.
But then came a game-changing breakthrough. Induced pluripotent stem cells, iPSCs. This really shifted the landscape, didn't it?
Dramatically. This was the Nobel Prize-winning work of Shinya Yamanaka. He discovered that you could take a fully differentiated adult cell, like a skin fibroblast or a blood cell, and reprogram it back to a pluripotent state, essentially turning back its developmental clock.
How? Just by adding specific factors.
Amazingly, yes. He identified 4 key transcription factors, octafer, SOX2, KLF4, and c-MYC, often called the Yamanaka factors. When these 4 factors are introduced and expressed in a differentiated cell,
They force it to become pluripotent again.
they induce a profound epigenetic reprogramming. The process involves widespread DNA demethylation, reactivation of silenced pluripotency genes, changes in histone modifications, essentially wiping the epigenetic slate clean and reestablishing the epigenetic signature of an embryonic stem cell.
What are the advantages of iPSCs?
Huge advantages. First, they can be generated from a patient's own cells, making them autologous. This potentially avoids immune rejection issues if they were used for therapy. Second, their creation does not involve the use of embryos, bypassing many of the ethical concerns associated with ESCs. They become invaluable tools for disease modeling. You can take skin cells from a patient with a genetic disease, make iPSCs, and then differentiate those into the affected cell type, like neurons for Parkinson's, to study the disease in a dish. Also used for drug screening.
Okay, sounds amazing. Are there any downsides or risks?
Yes, there are still challenges. One of the original Yamanaka factors, c-MYC, is a known oncogene, a gene that can promote cancer. Early methods often use viruses to integrate these factors into the genome, and the persistent expression of c-MYC, or the integration itself, raised concerns about tumor formation.
How's that handled now?
Newer methods use non-integrating approaches like using mRNA, proteins, or non-integrating viruses like Sendai virus to deliver the factors only transiently. They do their reprogramming job and then disappear, reducing the cancer risk.
Any other concerns?
Another area of active research is epigenetic memory. Sometimes iPSCs seem to retain some residual epigenetic marks from their original cell type. For example, an iPSC made from a blood cell might be slightly more prone to differentiate back into blood cells compared to an iPSC made from a skin cell. This incomplete reset could affect their differentiation potential or fidelity for certain applications, but the technology is constantly improving. #VC. Adult stem cells and the niche.
All right, let's turn to the stem cells that reside in our bodies throughout life, the adult stem
You can
cells.
do it!
Unlike
Let's work
ESCs
today
or
to make
iPSCs
tomorrow
in
a better
dish,
day!
these live in very
Okay.
specific
You
locations,
can
don't
do
they? The
it!
niche.
Let's work today
Absolutely.
to
The
make
niche
tomorrow a
concept is
better
critical for
day!
understanding adult stem
Okay.
cells. They
You
don't just
can
float
do it!
around, they reside
Let's
in
work.
specialized microenvironments within tissues. This niche provides a complex cocktail of signals, physical and chemical, that tightly regulates the stem cell's behavior.
What kind of signals are we talking about?
All sorts. Signals from neighboring cells, like Notch or Wnt, signals from the extracellular matrix ECM that the stem cell is touching via integrins, secreted growth factors, oxygen levels, even mechanical forces like tissue stiffness. The niche essentially tells the stem cell whether to stay quiescent, dormant, divide to self-renew, or divide and differentiate to produce progeny.
Can you give us examples of these niches and the stem cells they support?
Sure. We already mentioned the hematopoietic stem cell, HSC, niche in the bone marrow. It's a complex environment involving bone cells, endothelial cells lining blood vessels, mesenchymal stromal cells, all providing signals like CXCL12 and stem cell factor, SCF, to maintain the HSCs and regulate blood production.
What about other tissues?
There are mesenchymal stem cells, MSCs, also found in bone marrow, but also in adipose, fat tissue, and umbilical cord blood. They can differentiate into bone, cartilage, and fat cells, and their niche provides signals related to tissue repair and regeneration.
Even the brain has stem cells.
Yes, neural stem cells persist in specific regions of the adult brain, notably the subventricular zone, SVZ, lining the lateral ventricles, and the dentate gyrus of the hippocampus. Their niches support ongoing, albeit limited, neurogenesis, creation of new neurons.
And tissues with really high turnover must have very active niches.
Definitely. The intestinal stem cell niche, located at the base of the crypts in the intestinal lining, is a prime example. These stem cells, marked by the protein LGR5, are constantly receiving Wnt and Notch signals from surrounding Paneth cells and stromal cells. This drives the incredibly rapid turnover of the intestinal epithelium. The entire lining is replaced every 4-5 days.
Wow. Muscle regeneration, too.
Yes, skeletal muscle has resident stem cells called satellite cells, located just outside the muscle fibers. When muscle is injured, signals in the niche activate these satellite cells to proliferate and fuse to repair the damaged fibers.
So the niche is like the stem cell's command center. What happens if the niche itself gets damaged or changes?
That can have major consequences. Disruption of the niche, perhaps due to aging, inflammation, radiation, or mutations in niche cells, can lead to stem cell dysfunction. This might manifest as exhaustion. The stem cells can no longer effectively repair tissues. Or, conversely, aberrant signals from a damaged niche could potentially drive uncontrolled stem cell proliferation, contributing to malignancy. The health of the niche is critical for tissue homeostasis.
Okay, stem cells renew tissues, but all cell division, whether stem cell or regular cell, needs to be tightly controlled. This brings us to the machinery that governs the cell cycle and the process of programmed cell death, apoptosis. These are fundamental to maintaining balance, or homeostasis, #A. Cell cycle control and checkpoints, the rules of division.
Exactly. The cell cycle isn't just an on-off switch. It's a carefully orchestrated sequence of events. G phase, growth, S phase, DNA synthesis, G phase, preparation for division, and M phase, mitosis, division. And progress through these phases is driven by a core engine, cyclin-dependent kinases, CDKs, and their regulatory partners, the cyclins.
Cyclin-CDK pairs, like little molecular engines driving each phase.
That's a good way to think of it. CDKs are the engines, but they only run when bound to the correct cyclin partner. And the levels of different cyclins rise and fall dramatically during the cycle, ensuring the right CDK activity happens at the right time.
Okay, let's walk through the key pairs. What gets the cell moving from GO into S phase, committing to division?
That's primarily the job of cyclin D partnering with CDK4 and CDK6, often written as cyclin D CDK46. Their main target is a crucial protein called the retinoblastoma protein, RB.
RB, the famous tumor suppressor.
Indeed. In its active state, RB acts as a brake, holding on to transcription factors called E2Fs, preventing them from turning on genes needed for S phase. But when cyclin D CDK46 become active in GIA, they phosphorylate RB.
Adding phosphate tags changes its shape.
It does. Phosphorylated RB lets go of E2F. The freed E2F then activates the transcription of genes required for DNA replication, pushing the cell across the GS boundary. So phosphorylating RB is like releasing the parking brake.
Okay, brake released. What happens next in late GO and entering S phase?
Then cyclin E partners with CDK2. Cyclin E CDK2 activity ramps up in late GO. It helps with origin licensing, preparing the sites on the DNA where replication will begin. It ensures replication starts correctly.
And during S phase and into G,
Cyclin A partners with CDK2 and later with CDK1-CDC2. Cyclin A CDK complexes are important for controlling the progression of DNA replication itself and preventing the DNA from being re-replicated within the same cycle. They also help prepare for mitosis.
finally the big event, mitosis M phase.
Entry into M phase is driven by the activation of cyclin B partnering with CDK1, also known as CDC2. This complex, often called MPF maturation promoting factor, triggers the dramatic events of mitosis chromosome condensation, nuclear envelope breakdown, spindle formation by phosphorylating key proteins like nuclear lamins and histones.
So a sequence of cyclin CDK waves pushes the cell through the cycle. But it's not uncontrolled, right? There are regulators.
Absolutely critical regulators. There are CDK inhibitors, CKIs, that act as brakes. For example, the INK4 family, like P16, specifically binds and inhibits CDK46, putting a stop to GA progression. The CIPK family, including T21 and P27, have broader activity, inhibiting several cyclin CDK complexes, often acting later in GO or in response to stress signals.
And activators, too.
Yes, things like CDC25 phosphatases, which remove inhibitory phosphates from CDKs, helping to push the cell forward past certain thresholds. And CKAKE, CD activating kinase, which adds an activating phosphate. It's a complex balance of accelerators and brakes.
The real safety net, though, comes from the checkpoints. These are like quality control stations, aren't they?
Exactly. Checkpoints are surveillance mechanisms that monitor the integrity of key processes. If something is wrong, like DNA damage, incomplete replication, or misaligned chromosomes, the checkpoints will halt the cell cycle, preventing the cell from progressing with errors.
What are the major checkpoints?
A crucial one is the GS checkpoint, sometimes called the restriction point. This is where the cell checks for DNA damage before committing to replicating its DNA. If damage is detected, the master tumor suppressor P53 gets activated.
P53 steps in.
Yes, P53 acts as a transcription factor, turning on genes like the CKI P21. P21 then inhibits cyclin CDK complexes, halting the cycle in GRO and giving the cell time to repair the damage. The RB pathway is also critical here, acting as a gatekeeper.
Okay, what about during S phase itself?
There's an intra-S checkpoint. This monitors the process of DNA replication. If replication forks stall or the DNA gets damaged during copying, sensors like ATR and CHK1 kinases get activated to slow down replication and initiate repair.
And before the cell actually divides.
The GOM checkpoint. This verifies that DNA replication is fully and correctly completed before the cell enters mitosis. It ensures the cell doesn't try to segregate damaged or incompletely copied chromosomes. This often involves controlling the balance between the inhibitory kinase WEE1 and the activating phosphatase CDC25, which regulates CDK1 activity.
The final check seems incredibly important. The spindle assembly checkpoint, SAC, right in the middle of mitosis.
Absolutely vital. The SAC monitors whether all the chromosomes are properly attached to the mitotic spindle fibers before the cell initiates anaphase, the separation of sister chromatids. If even one chromosome is lagging or incorrectly attached, proteins like MAD and BUB send a wait signal, inhibiting a complex called the anaphase-promoting complex cyclosome, APCC.
Why inhibit the APCC?
Because the APCC is responsible for triggering the separation of chromatids and the exit from mitosis. By inhibiting it, the SAC prevents the cell from dividing until all chromosomes are perfectly aligned. This is crucial for preventing aneuploidy daughter cells ending up with the wrong number of chromosomes, a hallmark of many cancers. #B programmed cell death, apoptosis. So the cell cycle has checkpoints to ensure fidelity. But what if the damage is too severe to repair, or if a cell is simply no longer needed, like the webbing between our fingers during development?
And it needs to be eliminated cleanly. That's apoptosis or programmed cell death.
Exactly. Apoptosis is a highly regulated, active process of cellular self-destruction. It's often described as cell suicide. Critically, it's a tidy death. The cell shrinks, condenses its chromatin, fragments its DNA, and breaks into membrane-bound apoptotic bodies that are quickly engulfed by phagocytic cells. This avoids spilling cellular contents and causing inflammation, unlike necrosis, which is messy cell death from injury.
Why is apoptosis so important?
It's essential for normal development, sculpting tissues, eliminating structures, for tissue homeostasis, removing old or damaged cells, and for immune function, eliminating infected or cancerous cells.
How does a cell receive the signal to die? There are different pathways, right?
Two main pathways initiate apoptosis. The intrinsic pathway, also called the mitochondrial pathway, is typically triggered by internal cellular stress.
Like what kind of stress?
Severe DNA damage that can't be repaired, lack of essential growth factors or survival signals, hypoxia, low oxygen, or major problems with protein folding.
And how does that stress signal trigger death?
These stresses converge on the mitochondria. They lead to the activation of pro-apoptotic proteins from the BCL2 family, specifically BX and BAK. These proteins oligomerize, clump together, and form pores in the outer mitochondrial membrane.
Punching holes in the mitochondria.
Effectively, yes. This allows proteins normally kept inside the mitochondria to leak out into the cytoplasm. The most important one is cytochrome C.
Cytochrome C, isn't that usually involved in energy production?
In healthy cells, yes. But when it spills into the cytoplasm, it takes on a deadly new role. It binds to another protein called APAF1, forming a large wheel-like complex called the apoptosim.
The apoptosim.
Oh, yeah.
And that's the executioner.
It's the platform that activates the initiator caspase for this pathway, caspase-9. Activated caspase-9 then kicks off the final execution cascade.
Okay, that's the intrinsic pathway triggered from inside. What's the other route?
The extrinsic pathway, or death receptor pathway. This is triggered by external signals, often from immune cells like cytotoxic T lymphocytes.
How do they signal death?
They display specific death ligands on their surface, like Fas ligand, FASL, or TNF, tumor necrosis factor alpha. These ligands bind to corresponding death receptors, like Fas or TNFR1, on the target cell surface.
Receptor binding triggers death directly.
Binding causes the death receptors to cluster together, and this recruits adapter proteins like FADD, Fas-associated death domain, to the intracellular side. This complex, called the DISC, death-inducing signaling complex, then directly recruits and activates the initiator caspase for this pathway, caspase 8.
So, intrinsic pathway uses caspase 9, extrinsic uses caspase 8. Do they converge?
Yes. Both activated caspase-9 and activated caspase-8 lead to the activation of the downstream executioner caspases, primarily caspase-3, but also caspase-6 and Nemin-7.
Executioner caspase, what do they execute?
They are the real workhorses of demolition. Once activated, these caspases go on a rampage, cleaving hundreds of specific protein targets within the cell. They chop up structural proteins in the cytoskeleton, degrade proteins involved in DNA repair, dismantle the nuclear lamina, and activate enzymes that fragment the DNA. This systematic dismantling leads to the characteristic morphological changes of apoptosis and the tidy breakdown of the cell.
It sounds like a very controlled demolition. Is there a master switch deciding between life and death, especially a response to damage?
If there's one protein that often acts as that master switch, it's our friend P53 again.
P53 deciding life or death.
Yes. When activated by severe stress, like massive DNA damage, P53 can transcriptionally activate pro-apoptotic genes, including BAX. At the same time, it can repress anti-apoptotic genes like BCL2, so P53 can tip the balance decisively towards the intrinsic apoptotic pathway if repair isn't possible.
Are there proteins that try to stop apoptosis?
Yeah.
Inhibitors?
Yes, the cell has brakes on the death pathway too. The inhibitors of apoptosis proteins, IAPs, can directly bind to and inhibit caspases, preventing accidental activation. However, when the intrinsic pathway is strongly triggered, the mitochondria also release proteins like SMACDABLO, whose job is specifically to bind to and neutralize the IAPs, thereby allowing the caspases to do their work. It's another layer of regulation.
Okay, one related cell fate we should mention is senescence. It's not death, but it's not division either.
Right. Cellular senescence is essentially a stable state of irreversible growth arrest. The cell stops dividing permanently, but it remains metabolically active and often secretes a range of signaling molecules, the senescence-associated secretory phenotype, or SASP.
What triggers senescence?
It can be triggered by various stresses. One classic trigger is reaching the Hayflick limit, the point where telomeres, the protective caps at the ends of chromosomes, become critically short after many rounds of division. It can also be triggered by persistent DNA damage or strong oncogenic signaling, acting as a fail-safe mechanism against cancer.
And the mechanism involves.
Often involves the same pathways that cause cell cycle arrest, but made permanent. Activation of p53 leading to sustained high levels of the CKI p21 is one route. Another major pathway involves the CKI p16, which permanently locks down the RB protein, ensuring the cell can never pass the GS checkpoint again. So senescence is like putting the cell cycle into permanent park, whereas apoptosis is dismantling the car entirely. So having meticulously laid out the rules of development, the controls on the cell cycle, and the process of programmed cell death, we can now really appreciate what cancer is. It's the catastrophic breakdown of all these rules. It's development, but gone completely mad.
It's the corruption of those very same pathways we've been discussing. And the sources identify key characteristics that a normal cell needs to acquire to become truly malignant. These are the famous hallmarks of cancer. #A, hallmarks of cancer. Can you listen for us?
You can do
What
it.
capabilities must a
Let's
cancer
work
cell
today
achieve?
to make
Based on the
tomorrow a better
foundational work by
day.
Hanahan and
Okay.
Weinberg and subsequent updates,
You can
there's
do
a core
it.
set of capabilities.
Let's work today to make
First,
tomorrow a better
cancer
day.
cells need sustained
Okay.
proliferative signaling.
You can do it.
They have to generate
Let's
their own
work-
go signals or become hypersensitive to them, often by hijacking pathways like RTKs or Wnt. They don't wait for permission to divide.
Okay, constant go signals. What's next?
Second, they must evade growth suppressors. They have to disable the brakes, the tumor suppressor pathways like RB and p53 that normally halt proliferation.
So accelerator jammed on, brakes cut. Third,
Third, they need to resist cell death. They have to disable the apoptosis machinery, often by upregulating anti-apoptotic proteins like BCL2 or inactivating p53, so they ignore signals that would normally tell them to die.
surviving when they shouldn't. Fourth,
Fourth, they achieve replicative immortality. Normal cells can only divide a limited number of times because their telomeres shorten. Cancer cells bypass this limit, usually by reactivating the enzyme telomerase, which maintains telomere length, allowing them to divide indefinitely.
limitless divisions. Fifth hallmark.
Fifth, they induce angiogenesis. Solid tumors need a blood supply to grow beyond a tiny size. Cancer cells send out signals, like VEGF, to trick the body into growing new blood vessels into the tumor, supplying nutrients and oxygen. It's like building their own infrastructure.
Got it. Building a supply line.
Yeah.
And the sixth is perhaps the most deadly.
Sixth is activating invasion and metastasis. This involves acquiring the ability to break away from the primary tumor, invade surrounding tissues, enter the bloodstream or lymphatic system, travel to distant sites, and form secondary tumors. This requires processes like EMT that we discussed.
Right, the ability to spread. Is that all?
A seventh core hallmark recognized more recently is deregulating cellular energetics or metabolic reprogramming. Cancer cells alter their metabolism to fuel rapid growth. They often rely heavily on glycolysis, even when oxygen is present, the Warburg effect diverting building blocks for synthesizing new cells.
So 7 key capabilities they need to acquire. It's a multi-step process. #B, genetic roles and rules. How do cancer cells acquire these capabilities? It comes down to changes in specific genes. And we can classify these cancer-related genes based on their normal function, right?
Yes, broadly into 3 main categories. First, you have proto-oncogenes.
Proto meaning before. So their normal job is...
Their normal job is to promote cell growth and division in a controlled manner. Think of them as the accelerator pedal. Examples include genes encoding growth factors, receptors like HER2, signaling proteins like RAS, and transcription factors like MYC.
And in cancer, they become oncogenes.
Exactly. Through mutations, often gain-of-function mutations, they become hyperactive or constitutively on. It's like the accelerator pedal gets stuck down. This drives uncontrolled proliferation. Importantly, you usually only need a mutation in one copy, one allele, of a proto-oncogene to get this oncogenic effect. It's a dominant mutation at the cellular level.
Okay, oncogenes are the stuck accelerator. What's the second category?
Tumor suppressor genes. Their normal role is the opposite. They act as the brakes. They restrain cell proliferation, monitor DNA damage, and can trigger cell cycle arrest or apoptosis if things go wrong. Classic examples are TP53, P53, RB, and DNA repair genes like BRCA12.
And in cancer?
They are inactivated, usually through loss of function, mutations, or deletions. This is like cutting the brake lines. The cell loses its crucial inhibitory controls.
And the third category?
These are often called caretaker genes or DNA maintenance genes. Their normal job isn't directly controlling proliferation, but rather maintaining the integrity of the genome. They are involved in detecting and repairing DNA damage, fixing replication errors, ensuring chromosome stability. Examples include mismatch repair genes like MSH2 and MLH1.
So how does losing them contribute to cancer?
Loss of caretaker function doesn't directly make a cell grow faster, but it leads to genomic instability. The cell starts accumulating mutations much more rapidly because the repair systems are broken. This dramatically accelerates the rate at which mutations can occur in proto-oncogenes and tumor suppressor genes, speeding up the progression to full-blown cancer. They're like the mechanic who's stopped maintaining the car.
Okay, oncogenes, tumor suppressors, caretakers. Now, for tumor suppressors, there's a specific rule about how they need to be inactivated, isn't there? The Knudsen two-hit hypothesis.
Yes, formulated by Alfred Knudsen based on studies of retinoblastoma. Because tumor suppressor genes usually function recessively at the cellular level, meaning you need to lose both good copies to lose the function, the hypothesis states that two inactivating mutations, or hits, are typically required to eliminate the suppressor activity.
Two hits. How does that explain hereditary cancer syndromes?
In hereditary cancer syndromes, like familial retinoblastoma or Leif-Rhymiani syndrome, p53 mutations, an individual inherits one defective copy of a tumor suppressor gene from a parent. This is the first hit, and it's present in every cell of their body. This means they are born predisposed to cancer because now only one additional somatic mutation, the second hit, in the remaining good copy is needed in any susceptible cell to completely knock out the tumor suppressor function and initiate cancer. This is why these syndromes lead to much earlier onset and often multiple tumors. In sporadic, non-inherited cancers, both hits have to occur by chance within the same somatic cell lineage, which takes much longer. #C, hijacked developmental pathways, the betrayal. So bringing it all together, the central theme that emerges from the sources is profound. Cancer fundamentally involves the reawakening and corruption of ancient embryonic developmental pathways. The very same signaling systems that meticulously built the body during embryogenesis are hijacked by tumor cells to drive their own uncontrolled growth, survival, and spread. It's a betrayal of the developmental program.
Let's look at specific examples of this betrayal. We keep coming back to one, Katnin.
We do, because it's such a prime example. It's normal role, critical for cell fate decisions, axis formation in the embryo, and maintaining adult stem cells. Its hijacked role in cancer, particularly colon cancer, constitutive activation, usually due to loss of the APC brake. This leads to constant patentin signaling, driving relentless proliferation and maintaining a dangerous stem cell-like state in the tumor cells.
Okay. What about Sonic Hedgehog? Shh.
Normal role, patterning tissues like the neural tube and limbs, hijacked role. When constitutively activated, often via mutations in PASH or smoothened, it promotes tumor self-renewal, allowing cancer stem cells to persist and drives angiogenesis, helping the tumor build its blood supply. Inhibitors targeting this pathway are now used for cancers like basal cell carcinoma and medulloblastoma. Normal role, mediating cell communication for differentiation choices and boundary formation. Hijacked role, aberrant notch signaling in cancer promotes cell survival, prevents differentiation, enhances invasion and metastasis, and contributes to therapy resistance. It helps tumor cells adapt and persist.
So if you put these together, want driving proliferation and stemness, driving self-renewal and angiogenesis, notch promoting survival and invasion.
You can see how reactivating these core developmental signals provides the tumor with exactly the capabilities it needs to fulfill those hallmarks of cancer we talked about. The tumor essentially regresses to a pseudo-embryonic state. It taps into these powerful, primitive programs for growth and survival, but without any of the normal developmental constraints or boundaries. It becomes, as the sources suggest, like a miniature, corrupted embryo.
And perhaps the most dramatic example of cancer hijacking a developmental process is when tumor cells need to spread. This involves that epithelial to mesenchymal transition, EMT, we touched on earlier.
Yes. EMT is a fundamental process in normal development. It's essential for gastrulation, where germ layers form, for neural crest cell migration, for heart valve formation, wound healing. It's a normal, transient program where epithelial cells temporarily lose their characteristics and become migratory mesenchymal cells to achieve a specific developmental goal.
But in cancer, it's not transient, and the goal is metastasis.
Exactly. Cancer cells reactivate this entire developmental program, but often in a more sustained or aberrant way. The core molecular event is usually the downregulation and loss of E-cadherin, the glue holding epithelial cells together.
And what changes happen to the cell as it undergoes EMT?
It loses its polarity and tight cell junctions. It reorganizes its cytoskeleton to become motile. It starts expressing mesenchymal markers like N-cadherin, which promotes weaker, more dynamic adhesions suitable for migration, and vimentin, an intermediate filament protein characteristic of mesenchymal cells. It gains resistance to apoptosis and becomes invasive.
And this is all driven by changes in gene expression.
Yes, the whole process requires a profound transcriptional reprogramming. Key transcription factors, often called EMTTFs, like snail, slug, twist, and zeb, get activated. These factors directly repress E-cadherin expression and activate the expression of mesenchymal genes, orchestrating the entire transition.
The sources draw a powerful contrast here.
They do. The embryo uses EMT constructively to build tissues and organs. The tumor uses the exact same molecular program destructively to escape the primary site, invade, and metastasize. Its development literally running in reverse to achieve malignancy.
And this EMT process is also linked to the idea of cancer stem cells, CSCs, isn't it?
Very much so. EMT seems to be one way that cancer cells can acquire stem-like properties. The CSC model proposes that tumors are often hierarchically organized, much like normal tissues, with a small subpopulation of cells, the CSCs, that possess indefinite self-renewal capacity and are responsible for driving tumor growth, recurrence after therapy, and metastasis.
And these CSCs might share features with normal stem cells.
Yes, intriguingly, CSCs often express markers typically found in embryonic stem cells, such as OCT4, SOX2, and NANOG, the very factors that define pluripotency. This reinforces the idea that cancer involves a dangerous regression towards a more primitive, embryonic, self-renewing state, and EMT might be a key mechanism enabling that regression. So this deep dive into the shared mechanisms of development, renewal, and cancer isn't just academic. Understanding this shared vocabulary is precisely what has enabled the development of modern, targeted approaches to diagnosing and treating cancer.
Right. The knowledge translates directly into tools and therapies. Let's talk about diagnostic tools first. How does understanding these pathways help us detect cancer?
We can now look for the specific molecular fingerprints of these hijacked pathways. Techniques like fluorescence in situ hybridization, FIS, can detect gene amplifications, like the HER2 amplification in breast cancer. Polymerase chain reaction, PCR, and especially next-generation sequencing, NGS, allow us to rapidly sequence tumor DNA or RNA to find specific mutations, like an EGFR or KRAS, or gene fusions, like the BCR-ABL translocation that creates the Philadelphia chromosome in chronic myeloid leukemia, CML. These molecular diagnostics identify the specific drivers in a patient's tumor.
Identifying the driver then allows for targeted therapies. Let's review some key examples mentioned in the sources.
Okay. For CML, driven by that BCR-ABL fusion tyrosine kinase, the targeted drug imatinib Gleevec was revolutionary. It's a small molecule inhibitor designed specifically to fit into the ATP binding pocket of the BCR-ABL kinase, shutting down its aberrant signaling.
A true magic bullet for that specific cancer driver. What about for HER2 positive breast cancer?
For HER2 overexpression, the therapy is trastuzumab, Herceptin. This is a monoclonal antibody that binds specifically to the extracellular part of the HER2 receptor. This blocks its ability to signal and also flags the cancer cell for destruction by the immune system.
We also talked about epigenetic abnormalities in cancer, like silencing tumor suppressors. Can we target those?
Yes, this is a growing area. There are drugs that inhibit the enzymes responsible for aberrant epigenetic marks. For example, HDAC inhibitors, histone deacetylase inhibitors, aim to reopen chromatin and allow silenced tumor suppressor genes to be re-expressed. Similarly, DNMT inhibitors, DNA methyltransferase inhibitors, try to reverse aberrant DNA hypermethylation. These are used, for instance, in certain types of leukemia and myelodysplastic syndromes.
And moving towards harnessing the immune system.
That's been a huge breakthrough. Immune checkpoint inhibitors target proteins like PD-1 on T cells, or its ligand PD-L1, often on tumor cells, or CTLA-4, another break on T cells. These checkpoints normally prevent excessive immune responses, but tumors exploit them to hide from T cells. Blocking these checkpoints with antibodies essentially releases the brakes on the immune system, allowing T cells to recognize and attack the cancer cells more effectively. These have transformed treatment for cancers like melanoma and lung cancer.
And perhaps the most personalized approach,
No.
AR T cells.
Chimeric antigen receptor, CAR, T-cell therapy is remarkable. You take a patient's own T-cells, genetically engineer them in the lab to express a synthetic receptor, the CAR, that specifically recognizes a protein on the surface of their cancer cells, like CD19 on B-cell leukemias, expand these engineered cells to large numbers, and then infuse them back into the patient. These CRT cells then act like guided missiles, seeking out and killing the tumor cells with high specificity. It's shown incredible results in certain blood cancers. All these advanced therapies stem directly from understanding the molecular pathways, often developmental pathways, driving the cancer. Hashtag outro, final takeaway.
So our journey today has taken us from the intricate choreography of embryonic development through the resilient architecture of stem cell renewal to the devastating broken melody of cancer, where those same systems go awry.
And the sources leave us with a really powerful concluding analogy to tie it all together. They describe life beginning as this state of disciplined chaos. Think of the zygote, a single cell just humming with potential, capable of generating infinite fates, but tightly constrained and guided by that maternal blueprint and the unfolding developmental cascade.
Then, throughout life, stem cells are the living embodiment of that potential, keeping the melody of renewal alive, maintaining the harmony and structure of our tissues.
And cancer, in this view, becomes that same powerful, creative developmental song, but played way too loudly, erratically, refusing to follow the score, refusing to end. The tumor selfishly reclaims the potent self-renewal and signaling programs of the embryo,
You can
forgetting
do
its
it!
place in the larger
Let's
organism.
work today to
a
make
profound
tomorrow a
connection.
better day!
So the final thought for you,
Okay.
our listener, as you reflect on
You
all this
can do
intricate
it.
detail,
Let's
perhaps
work
consider
today
this.
to make tomorrow
If
a better
cancer
day!
represents a reversion
Okay.
to a corrupted
You can
embryonic
do
state,
it!
if
Let's work
malignancy requires reactivating these primitive developmental programs, could the ultimate therapeutic strategy involve not just trying to kill these cells, but somehow forcing them to finish the developmental program they've restarted, forcing them to differentiate, to mature, to lose that dangerous immortality and plasticity?
That idea, shifting the focus from cytotoxic killing to differentiation therapy, is really at the heart of leveraging our understanding of development to fight cancer. It suggests that the roadmap for treating the disease might be found within the very blueprint that builds life in the first place.